Good morning again, everyone, and thank you for joining. I know we're a small group, but hopefully we'll make the best of it. Today I will be talking to you all about my experience working on the Analyze Med project through the Laboratory of Applied Informatics Research here at UNC with Dr. Javed Mustafa and my colleague Jeffrey Stocker. So what exactly is the Analyze Med project? Well, to get a better understanding of this project and its purpose, it is important to have a general idea of how our bodies respond to different medications. So as you can see from the picture on the left there are varying degrees of response to drugs in humans. For the most part, most people will elicit a full response, which is essentially, as clinicians and pharmacists, what we want. We want people to have positive treatment outcomes, have minimal to no side effects, and that's essentially our goal. But we find that, there are some patients who, either elicit only a partial response, or sometimes they don't respond at all. Or worse yet, there are people who will respond to a medication negatively and show very severe adverse reactions. So why is there such a huge degree of variation is the question. And the answer to that is there are actually many different factors that can influence the pharmacokinetics, pharmacodynamics, and the overall response to medications in our bodies. As you can see to the figure on the right, all of those are just some of the many different things that can affect this phenomenon in our bodies. Things as simple as just how old we are, our gender, our weight, to as complicated as our renal function, our liver function, other medications that we take, other diseases that we may have. All of these things can have positive and negative effects on drug response in our bodies. So one key factor that is now being examined as a huge influence on our responses to medications is our genes and overall genetic makeup. And that is where pharmacogenomics is starting to play a huge role in healthcare and informatics. So what is pharmacogenomics? It basically is the scientific discipline that understands how genetic variability affects drug treatment outcomes. This science continues to play an integral role in clinical decision making. And we are seeing that, as we're gearing more towards personalized medicine, precision medicine, that genomic data is slowly beginning to be integrated into health IT systems and EHRs. And so the Analyze Med project that we worked on not only provides a way to utilize this information and research, but serves as a stepping stone towards this whole new transformation that we're seeing in the field. So the Analyze Med project is an ongoing pharmacogenomics endeavor that seeks to gather a deeper understanding of the potentially adverse interactions between genes or polymorphisms and different prescription medications and other drug molecules. For the focus of our project currently, we decided to specifically concentrate on the African American patient population. The main objective of this project is to create different links between the following information: the medications and or drug molecules, the genetic variants, mutations, or polymorphisms, the adverse reactions, and its related literature and publications found on PubMed. Now, this data is collected and then compiled into a database that is both searchable and easily accessible to its users. As the system develops, it ideally will be linked to other health information systems, such as EHRs, which then can be utilized for clinical decision support during the input of medication orders by healthcare providers. In addition, this database can be used by researchers and patients to retrieve information about different genetic polymorphisms, their relationships to medications, and the subsequent adverse effects that occur. Our work on this project so far can be divided into three main stages. The first being the actual creation of the database, second, data collection, and finally making improvements to the database interface and its features. So this past summer, my colleague, Jeffrey Stocker, actually started to work on the AnalyzeMed project, and he actually laid the foundation for the creation of its web application and database. Even though I joined the team after much of this work had concluded, Jeffrey provided a brief overview of the concepts and steps that he used during this process. After conducting some preliminary research on pharmacogenomics in the African American population, the ER diagram seen on the right here was created as an initial step towards database design. This diagram was then used to conceive the relational mapping in schema, which provided an outline for what ultimately became our final product, the AnalyzeMed Pharmacogenomics database. By employing Python with a Flask framework, the web application was built using the PyCharm program, and this enabled us to use SQLite for actual database curation on the back end, while HTML and CSS concepts were applied to generate the visual aspects and other front-end features of the web application. The main Python file that we created basically ties both the front end and back end features of the application together. And anytime we needed to make updates to the database or its web application, we did it by changing pertinent sections of the code as needed. So after joining the team this past fall, my assigned tasks included conducting a literature review, collecting data, and providing insights and recommendations for improvements to the user interface and other database features. So to begin, I started obviously with the data collection component and the literature review. To construct a pharmacogenomics database that would be most beneficial for clinical decision-making and research purposes, I conducted an initial cycle of data collection over a 4 to 6 week period, primarily using PubMed. I performed a literature review to collect the following pieces of data: the molecule or drug name, the associated genetic polymorphism, the relevant ICD-10 code, a description of the adverse effect, and a link to the supporting literature. The figure to the right there shows the number of articles that I retrieved using key search terms that included African Americans, pharmacogenomics, genetic variant, and genetic polymorphism in various combinations. And additionally, I even used an advanced search using mesh terms to potentially help me refine the search results further. In order to actually figure out what articles and data would fit the purposes of our project, I did a quick review of all the titles and abstracts of the articles that were generated on PubMed. And based on that preliminary screening, I then started creating a list of potential articles that we would be able to use. And then from that list, I basically went through each and every article, did a thorough analysis of each in order to be able to gather the data that we were looking for. Articles that did not discuss the African American population and articles that didn't contain a specific molecule polymorphism pair were excluded from our data set. And in addition, articles that did not have full text accessibility were also excluded from the list, as this limited our ability to establish clinical and or statistical significance for a given drug-gene interaction. Furthermore, an effort was made to include the most recent studies to ensure the collection of the latest pharmacogenomics data and research. So once a final list of supporting literature was created, I manually went through each article to extract the data that we were looking for and stored it in an Excel spreadsheet. And then at the conclusion of my data collection step, the spreadsheet was converted into a CSV file, which then was imported into the Analyze Med project file in our PyCharm. And then we had a Python file that was specifically coded in order to add data to the database. And so this file was imported in there to be able to collect and update the data into our web application and database online. So the figure on this slide will show you all the data that is currently included in our database. And this is really all the data that was collected during the data collection step of this project. So the last 4 weeks of the internship experience was dedicated to improving the user interface and other database features. The following are ideas that I had proposed as possible changes and or additions to the web application and database. So one idea was to improve the overall visual appeal of the web application and database. I also thought it would be beneficial to have a filter and sort feature of some sort on the results page, which would basically allow us to display results by different categories, such as molecule name or polymorphism or the most recent study. I also determined, just kind of using my clinical perspective, that it would be beneficial to be able to search the database more generally by, say, for instance, drug class or some other keyword, so that you know, in the real clinical setting, sometimes you'll come across situations where, you know, you may not be looking for a specific drug or molecule in particular to retrieve information. And so instead of, you know, typing in atenolol, which is a blood pressure medication, and you can type in like antihypertensives, and the system should be able to generate pretty much all of the blood pressure medication data. So, you know, an example like that would definitely be beneficial to our database. The other one, again, which would be beneficial to clinical practice, would be having information on the database that would contain recommendations that would be helpful during clinical decision making. So sometimes when you're looking through the articles, they will have specific recommendations about dosing or about checking renal function or something like that, which, you know, is important to keep in mind when we're trying to use these medications in our patients. So, you know, having that information available on the database would also be very helpful. And then one feature, again, that kind of came from my clinical experience, was to have a feature that allows a clinician or patient to input a patient's medication list and retrieve all relevant results based on that. And then finally, devising a way in which the web application can be connected directly to PubMed and be able to conduct searches to retrieve supporting literature and automatically extract data to input into the database, was one of the last ideas that I had come up with. So unfortunately, due to time constraints, it was not possible for us to fully develop all those ideas that I just listed, but we were able to successfully implement a feature that allows us to search the database by using drug names from a medication list, excuse me. This was done by adding additional code to the main Python file in our program, as shown in the figure below. So the main Python file here that you guys see is actually much longer than what's visible here on the screen, but I just wanted to point out where I had actually gone in and added this search feature. So if you look here, basically what this code says is that, you know, a user will put in a search criteria of some sort. So that's what we've identified as search string here. And then they're able to select, you know, what the search parameter is. So what we did was we added an additional search parameter here titled medication list. But for this, because we are potentially entering more than one molecule there, the system needs to know to look at each of them. So that's how we determined that we would need to put in a while statement with a for loop, so that, you know, the system knows to check each and every drug that is inputted there. So that is the main difference between the code there compared to the other search parameters. And that feature was one of the last sort of major updates that we made to the database. So I would like to share with you sort of our final product. Everyone actually can access this. If you go to our project page on the UNC website, analyzemed.unc.edu will get you to our project site. And if you click here, this is our actual web application. I'm sorry, is everybody able to see that?
Yes, we can see it.
Okay, great, thank you. So this is really what you see now when you access our application. So this is the main page, and this is where you can go in and search. So, you know, you can search by a specific polymorphism. Like if you're a researcher and you know, like, hey, this is a variant I want to look at, you can search by that variant by, you know, using the search parameter. If, you know, you're looking at a specific illness, or you could search by like ICD-10 code, and that'll give you some results as well. Or, you know, let's say you're looking at, you know, ones that require lower dose, you can put description, select description of effect and search lower dose and hit search, and it'll give you ones where the description of the interaction is a lower dose requirement. You can obviously search by the molecule. So let's say you want to find out if there's any relevant research about tacrolimus, which is an immunosuppressant. And we find that there is one currently in our database, which gives you the polymorphism, what happens, ICD-10 code, and then there's a link to the PubMed article here where you'll find more details about this drug-gene interaction. And then the one that I'm proud to contribute is the medication list feature. So here, let's say you have a patient that comes to you, you can put in multiple medications here, and let's say that is our list, and it will generate all the ones that are relevant to all of those medications in our results page. So that kind of gives you a little preview of what we have created. So let me just get back to our slides here. And feel free to explore that. Again, you can access it through analyzemed.unc.edu and do check that out. So one of the other things that we started working on towards the end of the project, and is still sort of a work in progress, is we actually started working on the initial steps towards creating a connection between the AnalyzeMed web application and PubMed by utilizing the BioPython package and its Entrez database available through Python. This is definitely something that both Jeff and I sort of had to self-learn, but this is sort of the initial work in code that you can see where we've basically establish that connection. We're hoping, as future interns take over the project, that they'll be able to complete the additional steps that will enable direct information retrieval from PubMed into our database. So moving forward, we expect that the data collection process will continue as more pharmacogenomics research develops. Additionally, to add more value to our database, it is expected that the database will include data from other ethnic groups in the future. We also hope to devise a way for future project members to have an easy way to access and manipulate the web application and its database via the chip server. We will essentially like to change the look and feel of the application and database to make it more user-friendly and create varying interfaces and features for different users. Basically, that means that, right now, anybody and everybody can kind of go into the database and kind of use it the same exact way. But essentially what we would like to do is kind of have sort of an introduction page where, it will prompt and ask whether you're a clinician, are you a researcher, are you a patient? And based on that, you will have different features and views available to you. So, for instance, a researcher or a researcher may not need the medication list search feature, whereas a clinician or a patient will. And so that will be accessible more for them versus a researcher. And so, things like that, we would kind of like to streamline and make specific for different users. So that's definitely something down the road that we would like to work on and see improved. And then once that is done, we would also like to test this database and get feedback on its usability and additional improvements that can be made. And then, as I mentioned, direct information retrieval from PubMed and automatic uploading of data is definitely a goal. And then finally, we do anticipate that the AnalyzeMed pharmacogenomics database will be a useful tool that can be integrated into AHRs and health IT systems to assist with clinical decision support and improving overall health outcomes. So, in conclusion, working on the AnalyzeMed project was truly a rewarding experience, and I would like to thank Dr. Mustafa and Jeffrey again for giving me this opportunity. The internship experience really allowed me to help create a foundation for what we hope will be the future of pharmacogenomics and health IT, the project really allowed me to use both my informatics knowledge as well as my pharmacy and clinical background to really contribute to the project. I was able to refine my literature review, data collection, and Python skills. And from a clinician perspective, I was able to propose changes that I hope were valuable and beneficial to clinical practice and research. There were a few challenges along the way. There was a slight delay in transition and onboarding of some of the technical aspects for the project. But in hindsight, I think that also the delay allowed me to focus a little bit more on the literature review. So I guess it was good in a way. And then there were definitely some concepts that I had to self-navigate and learn. But I think that was great because now, I, for example, know how to create a web application using Python and Flask. I know about Entrez and BioPython and how to create links between, our program and those databases using Python. So I've definitely learned a lot of wonderful things through this experience that, I we've only sort of touched on in some of the courses in the program. So I realized that, you know, this process of learning and developing my informatics knowledge and skill set will continue, especially as the technology continues to change and the world of informatics grows. But I look forward to being a part of even, you know, many more initiatives that continue to add value to healthcare technology and improve patient care and support clinical practice, just like our Analyze Med project and its pharmacogenomics database. So does anyone have any questions about Analyze Med?